Today we'll be looking at gene and gene action. So we are going to be looking basically at additivity or additive gene action, dominance, and epistasis. Then we'll also look at some other minor gene actions which are fallouts from these three basic ones. So as we know that all genes are the unit of inheritance or the hereditary materials, though located on the chromosomes, are transmitted from parents on to their offsprings. And these genes are located at particular locations called the locus. In plural, this is known as the loci. And they are made up of nucleotides, which are components of the DNA. So the parts of the genes found at a nucleus are referred to as the alleles, and these alleles could be homozygotes or they could be different, in which case they are said to be heterozygotes. Now the alleles are the alternate forms of the genes, and we usually use letters to represent the different alleles of a gene. Now genes usually appear as discs or are represented as bands when they are viewed on the chromosomes, when they are viewed using electron microscope. The action of the gene on an individual animal can be complementary, in which case we say it is passive, or it can be antagonistic or negative. At the same time, it can also be neutral. So genes can complement each other, they can antagonize each other, and when they are in a neutral state, it means that we could say that they are not bothered by the action of the other gene. Now, there are basically three types of gene action, and we'll be looking at them one by one. The first one is what is known as additive gene action. We also refer to it as additivity. Now, additivity occurs when each gene that affects a phenotype of an individual makes a specific contribution to a trait, and then the sum total of which gives the total phenotypic value of that individual for the trait of interest. So what this means is that every gene adds something to the phenotypic value of a trait. This addition could be positive, which means it is complementary. This addition could be antagonistic, which means it is negative, and this addition could also be neutral. So let's consider a character, for example, that is governed by a single gene locus with two alleles, capital A and small a. And these have been measured to contribute 1 and 0 to the total phenotypic value of an individual, respectively. So in this case, then we expect that the phenotypic value of the character in question that the individual is carrying should be according to the following genotype. For example, if the individual is carrying AA, the additive genetic variance here is going to be 1 plus 1, because capital A, is measured to have 1. So 1 plus 1 will give us 2. And if the individual is heterozygote, it is 1 plus 0. 0 because little a is measured to contribute only 0 to the phenotypic value. So 1 plus 0 will give us 1. While if it is in the recessive state, we know that is 0 plus 0, which is equals to 0. One interesting thing from this example, notice that the phenotype A small a, that's the heterozygote, is exactly midway between both homozygotes. So quantitative traits, however, are controlled by many genes, making this example to look very simple. And each of these genes exerts its own effect or influence on the final phenotype. And the influence could be, as we said earlier, complementary, could be antagonistic, and it could be neutral. So, to obtain the phenotypic value of such traits, that would mean the sum total of all the individual effects or the contribution of all the genes that are controlling the traits. So let's consider this example. For example, we have a trait that is governed by 4 different gene pairs: small a, capital B, small b, capital C, small c, and capital D, small d, with measured additive effects of 2/0 for this, 1/0 for this, 3/0 for this, and 2/0 for this, respectively. So if we want to know what is the phenotypic value of the trait, if this is the combination of the genes. So from this example, for A, small a, it will be 2 plus 0. B, small b, it will be 1 plus 0. C, small c, will be 3 plus 0. And then D, small d, will be 2 plus 0. And if you add up, this will give us a phenotypic value of 8. Here we'll see that the dominant alleles are all contributing positively to the final phenotypic value. So they complement each other. Meanwhile, we can see that the recessive alleles are neither antagonistic nor complementary, rather they are neutral. So they are not contributing anything, but they are also not directly affecting the final phenotypic value of the animal. But we have situations in which certain genes may exert an antagonistic effect. Like in the example below, the example we just used, let's assume that this gene C actually had a phenotypic contribution of 3/-2 as its additive effect. Then we'll realize that the phenotypic value of the trait will change. Indeed, it will become, for capital A small a, 2 plus 0, 2. For capital B small b, 1 plus 0 equals to 1. And then for capital C small c, it will now be 3 plus minus 2, which will give us 1. So it's beginning to show a negative effect, and this will affect the final phenotypic value, reducing it to 6 rather than the 8 that we saw in the previous example. So we want to extrapolate this a little bit by looking at skin color in animals. And skin color in animals is controlled by additive gene action, such that animals with this genotype show white coloration, and those with this or this are light in color. Those that have this genotype are medium in color, showing the effect of the dominant alleles. And those with this are dark in color, while those that are having double homozygotes are completely black in color. So we see that the effect of the recessive gene leads to reduction in the magnitude of the color of the animal. So let's take another example to see. We want to assume that fleece weight, we're not looking at color in this case, fleece weight in sheep is controlled by two pairs of genes, that's capital A small a and capital B small b. And each of the genes, capital A small b, adds 0.1 kilogram to the basic genotype value of 5.0 for this allele. It means that the initial level is of a small b small b is 5.0 kilogram of fleece weight. Now the genes small a or small b add nothing to the phenotypic value. So it means this is the basic line or the baseline, and it will produce for us 5.0 kilogram of food. Also, we must take into consideration that this example is when the environment is assumed not to have any effect on the expression of the genes. So when a dominant allele with this shape is mated to the basic one, you recall that capital A and capital B adds 0.1 to the basic weight. It means that animals that are carrying this genotype are going to have 0.4 more kilogram added to the fleece weight compared to the basic line, which is just 5.0. This as a result of 0.1 plus 0.1 plus 0.1, so that means 5.40 kilograms. So in the F1 progeny, which is capital A small a, capital B small b, the phenotypic value of that one is 5.20. And what that simply means is if you add this together and divide by 2, you get 5.0. Or if you remove 1a and 1b, capital B each, it will also give you 5.2. So we want to know what happens when these F1 progenies were crossed together. And the result for the F2 generation are these. So these are the genotypes that were obtained. And if you now factor in the contribution of the capital A and the capital B genes, which is 0.1. So all animals with this genotype, we have 4 times 0.1, which means 5.4. All animals with this genotype, we have 4 times 0, 3 times 0.1, 3, 1, 2, 3. This small b is neither antagonistic or complementary. It's adding nothing. It's at 0 to the baseline. So this will be 5.3. And as you carry out those substitutions, you notice a reduction. And when there's an increase in the dominant gene, there's also an increase in the fleece weight. So in this particular example, 64 animals were evaluated, and 4 of them had this genotype, 8 had this, 16 had this, 4 had this, and these were their calculated fleece weight. And when we took an average of the entire thing, we notice an average of 5.2, which is similar to the phenotypic value of the F1. So this example clearly shows or highlights two very important points. And these are that one, the mean of the F2 is 5.2, which we observe is the same as the mean of the F1 generation. And the second thing here is that in the F2, we observed a greater deal of coloration, which has appeared, which shows that various animals were able to fall into the various classes of fleece weight as compared to at the F1, where all of them were just at 5.2 kg. So we had as low as 5.0 and we had as high as 5.4 kilogram fleece weight. It should also be noted that we'll get a wider range of variation if environmental factors were allowed to have their way. In the example we just observed or looked at, environmental factors were held. It was in a closed, controlled environment. If these animals were to be exposed to a physical environment, the variation is likely to be wider than this. So that is additivity. The next thing we want to look at is dominance. Okay, so before we go further, I want to remind us here that additive genetic variance is transferable from parents onto their offspring. Dominance, on the other hand, is an interaction that occurs between alleles at a locus. And dominance is a case where a gene is able to suppress or is able to mask the expression of its partner, which in this case is said to be recessive. And this happens for genes that are alleles, that is, they somehow carry similar letters. They are also found on the same gene locus. So one important thing about dominance is that it is not transmitted from one generation to another, unlike additive genetic effect. Dominance happens just at the point when the various alleles are interacting at a particular gene locus. And there are 4 types of dominance effect. And these are, we have what is known as lack of dominance or co-dominance. So in this situation, what happens is that neither of the genes is able to dominate the other, such that both genes are able to express themselves in the phenotype. So a typical example of this is found in the wrong coat color of cattle, where when a red cattle is mated with a white colored one, uh, neither of the two colors, red or the white, is able to dominate each other, such that in the heterozygote animal, both colors are shown in the coat of the animal. The second type of dominance effect is what is known as complete dominance. And when this occurs, what is noticed is that both the heterozygote and the dominant homozygote tend to have the same phenotypic value. You can't actually differentiate the dominant homozygote from the heterozygote. But in this case, the recessive gene is completely deprived or prevented from showing itself in the phenotypes. So the third type of dominance interaction is what is known as incomplete dominance or partial dominance. And in this case, what happens is that the phenotypic value of the heterozygote will lie somewhere between the values of both the dominant homozygote and the recessive homozygote. But where it is lying is not usually at the midpoint. So the implication here is that the dominant gene is not able to completely mask the expression of the recessive gene. And in this case, unlike incomplete dominance, 3 different individuals are produced in the F2 that are easily distinguishable from each other. So in animal, an example could be taken from in Hereford cattle. In this cattle, the presence of this gene, capital C, will cause the individual to be dwarf. Then the small c will result in a normal individual, while the heterozygote results in a phenotype that is midway between the normal and the dwarf. So because of incomplete dominance, we have 3 classical distinguishable phenotypes. Meanwhile, in the case of complete dominance, we observe that we can only have two basic phenotypes because of the dominant effect of the dominant gene. So this is an example of incomplete dominance in a plant species. So we can see that we have this, this, and this due to the effect of incomplete dominance. Then the final one is known as overdominance. And overdominance occurs when the phenotypic value of the heterozygote is superior. Is so superior that it's better than either of the double homozygotes, either in the dominant state or in the recessive state. So this sometimes happens when animals are crossbred and they exhibit heterosis, or what is known as hybrid vigor, that is more than 100%. Then we have a case of overdominance. So an example of overdominance can be found in the sickle cell anemia in humans, where the SS will suffer from anemia, and those carrying the AA genotype suffer greatly from malaria. But surprisingly, the AS heterozygote rarely dies from malaria, or indeed show cases of anemia. So this is a classic case of overdominance above the two, the SS who suffer from anemia and the AA that suffer from malaria. So this is possibly as a result of the exhibition of heterosis in AS heterozygote. So this is an illustration showing three of the different types of dominance. This is complete dominance. You see that the heterozygote and the dominant homozygotes have similar phenotypic value. In the case of incomplete dominance, the heterozygotes are midway in between the dominant homozygote and the recessive homozygote, but not at the midpoint. And in the case of overdominance, we see that the heterozygote is the phenotypic value is much greater than that of the homozygote dominant and the homozygote recessive. So these are the four basic types of dominance. The next gene action is known as epistasis. And this is a non-additive gene action. Just like dominance, it is not transferable to the offspring. And this is due to interactions between genes that are non-allelic, that is, genes that are not found on the same gene locus. And these kind of genes are usually expressed by the use of different alphabets. So the gene at one locus that dominates the gene at another locus is said to be epistatic. And the gene that is dominated is said to be hypostatic. Now, the type of epistasis depends on the gene effect at each of the interacting loci and the number of loci that are involved. So there are many types of epistatic interaction, but that is not the focus of this lecture right now. Now, pleiotropy is a unique type of gene effect. It's not among the basic three types that we have looked at, but it's a special type of gene effect. And in this case, what we have happening here is one gene is shown to have different effects on several characters at the same time. And this phenomenon is known as pleiotropic effect or pleiotropy, and this is the chief cause of what is known as genetic correlation between characters. And where there is genetic correlation, it leads to what is known as correlated responses in traits. So genetic correlations is dealing with correlation between traits at the gene level. So correlated responses could be either positive or they could be negative. So we want to consider a situation where a gene, capital A, is exerting its effects on 4 different traits, X1, X2, X3, and X4 at the same time. So it also influences how these traits, where there is correlation between each of the traits. So if, for example, there's a single deleterious gene which results in a number of abnormal characters inherited as a single package, where you have such a situation where one gene results in different abnormal characters, but these are all inherited as a result of its manifestation, then we refer to such a syndrome as a pleiotropy effect. So this is an illustration of an example. Our gene A is affecting character X1, X2, X3, and X4. And it's not only affecting them, but also how they are correlated with each other. X1 correlated with X2, X1 correlated with X3, X1 correlated with X4, and so on and so forth. So when a gene has this power, it is exerting a pleiotropy effect. Finally, we want to look at penetrance and expressivity. So penetrance is actually the ability of a gene or a trait to express itself in a population. So when a gene or a trait expresses itself in a population, we say that it has penetrated that population. So where a gene fails to express itself in a phenotype, even though it is present, we say it is that its penetrance in this case is very low. Because of the lowness, that is why it will not be seen in the population. An example of such is found in polydactyl. This is a condition that is common in humans and is also found in several animals like the chicken, and is manifested in the appearance of an extra digit or finger or toe, and this is controlled by a dominant allele. Unfortunately, there are not all people that are carriers of this gene. We especially show the extra digit in the hands or the foot. So in this case, we say that the penetrance is low or it has not fully penetrated the population. When penetrance is complete, the gene or the genes for the trait will be fully shown or be fully expressed in the population. So penetrance could be incomplete, in which case we say it is reduced. Also, where in this particular situation, the traits will be expressed in some individuals in the population and not in some individuals. And can be in this reduced penetrance is influenced by factors such as genetics, and environment, and lifestyle that are yet to be fully identified. So many developmental traits sometimes fail to show penetrance. At times, they show variable patterns of penetrance, either very mild, intermediate, and severe. Of course, when they are severe, they are found everywhere, easily found. So expressivity is the extent to which a gene is expressed in the phenotype. That is the extent to which the gene has penetrated that particular population. Uh, so it's also viewed as a variation in phenotypic expression when an allele is fully penetrant. This also depends on the genotype and certain external environmental factors. It could also be complete or reduced. So when we look at the polydactyl example, individuals will have full-size extra digits, in which case we say there's complete expressivity. When the extra digits are shown, it's an example that there's full penetrance and it is fully expressed. Or it could just be a stop, in which case it is not fully expressed but variably expressed. So genetic, environmental, and lifestyle factors also affect penetrance. So this is an illustration to show penetrance and expressivity. We are taking an example of a disease condition. So we can see in this first one that there's complete penetrance because all the individuals are showing signs of the disease. Here we're having variable penetrance because some of the individuals have the disease while these other people are not showing it. And here we are trying to see this is variable expressivity. The disease condition is expressed in some individuals and not expressed in some others. This is both variable penetrance and expressivity, penetrance, expressivity. Okay, and this expressivity, sorry, and the normal individuals are not carrying the disease condition. So the condition has not penetrated, and as a result, it is not expressed in the population. So these are examples of gene actions that are common in animals, and I know that they are also workable in plants. And with this, we have come to the end of this module. Don't forget to like. You can share a comment so that we can interact. Please feel free to share with a friend and subscribe so that you get our next video. Thank you and have a great day.